Alright, so we're on our last section, part 3 of Module 9. So this is the last lecture for this week's lecture series under Module 9 when we are focused on taxonomy and systematics. Make sure the readings, make sure you're doing those 147 to 165. And so this will be the last lecture that your quiz is covering that you'll be taking next week. Okay, so just again, your assignments, make sure you're doing those. I also sent out an email with everything that is due, and I put it on Cobra. So hopefully you've been notified in some way, shape, or form of what's due so you can keep on task. Also, you can look at your calendar in Cobra, that works too. Make sure you're turning in your presentations, the topics only. That's due April 10th, so post your choice under the Module 8 discussion thread. First come, first serve. Also, due next week, on Tuesday, April 14th, is your Module 9 quiz. So everything that we covered this week, the lab, all 3 lectures, those are all due on Tuesday by 11:59. Make sure you study for that. It's open book, however, 20-minute long, 10 random questions, so you should do okay. You guys did pretty good on your last quiz. So also due is lab number 9, communicules. That's due on Tuesday, April 14th, so make sure you finish that up and get that turned in. A lot of you have been turning in Word documents that you type up. That works perfectly. I appreciate that. That way I don't have to decipher any handwriting. So if you prefer to do that, we do have the updated Communicule Lab on Cobra, so you're able to download it virtually as well. And then lastly here we have our Evolving Switches video due. It's only 15 minutes. There's a quiz that goes with it. You can find it under your quiz section. There's only 9 questions, but it's all fill in the right in the answer, so you'll have to do a little bit of typing. So, but it's pretty much open book or open video, I guess. So, and I don't believe that there's a time limit on it, so that should be pretty easy as well. And that'll help prep us for next week's lab and lecture as well. So today we're covering the last little section here of Module 9. We're going to go over molecular clocks and horizontal gene transfer. So today's lecture is going to be pretty short and sweet. I only have a few slides and then a short review, so hopefully since I know a couple of my other lectures ran a little bit long, you won't have to sit through one that is that long for today's particular lecture, especially since the weather's getting so nice out. Okay, so when it comes to molecular clocks, there is a theory here that I'd like to talk about. So this is what we call the neutral theory of evolution. So what this basically means is that molecularly here, favorable mutations that we have in our genetic code are pretty rare. So something that makes you survive really, really well is typically hard to find. And mutations that we have that are harmful or detrimental are typically quickly eliminated. So if you have some sort of gene that inhibits your survivability, typically you're picked off by a predator, or you're not able to find food, and you are eliminated from the gene pool. So those harmful mutations in your genetic code are typically eliminated pretty quickly. So what this is trying to get at is this theory is saying that most of the mutations that you see in your genetic code are neutral mutations. So they don't cause any sort of benefit, but they don't harm you either. So that's what this theory is talking a little bit more about. So if we consider how often mutations are happening, they typically, if we consider that they're happening at a constant rate, then they can be used to measure evolutionary time. So if you pretend that mutations are happening an equal number of mutations in every generation, say there's 100 billion different mutations that are happening each generation. So they're pretty spaced out. They're pretty constant. And so that way we know that if that's happening, we can measure time a little bit better. That's happening over the course of all of time. So we can measure, how evolution is happening and how frequently it's happening, and how old or how young a species is based on those neutral mutations. So one important thing to note is that these mutations are not perfectly linear over long periods of time. There are incidences of mutations happening more frequently, typically due to generation times. There's different generations that are occurring. There's possibly different environmental factors that are taking place. And then also, not all organisms are evolving at the same rate. Some typically take longer, where others evolve a lot more quickly. Like you would see that happening in a species that reproduces fairly quickly, like Drosophila, which is your fruit fly, whereas species like humans here take a longer time to have these mutations occur because they're not evolving at the same rate here. Okay, so this is just a nice little visual for you to look at and see how this neutral theory of evolution is occurring. So if you see in this image here, we'll only talk about two. We're not going to talk about nearly neutral theory. So let's talk about the top two. We'll talk about selection theory, which we've covered in past modules, and we'll cover neutral theory, which is what we're talking about right now, that is new to you. So when you have selection theory, so if you're talking about genetic selection, typically we either see harmful mutations that are occurring or advantageous mutations that are occurring. So when you see in selection theory, you see a lot more harmful mutations that are happening, because those individuals are either picked off by predators, are not able to survive, serve as food for another organism. But the advantageous group, or the advantageous population of genetic individuals here, they typically will survive and go on to procreate in the future. So that's our selection theory. Now the neutral theory is a little bit different. We have another large amount of harmful genetic mutations that are typically not going to survive here, and we have a very small amount of advantageous genetic mutations that are occurring, very small. And then we have a pretty decent sized amount of neutral genetic mutations, which aren't helping an organism survive, and they're not harming an organism and causing an inability to survive. So we'll have either those neutral genetic mutations or the advantageous ones survive, where the harmful ones will be picked off. But the important thing to note here is that these neutral genetic mutations can help us place these organisms on an evolutionary timeline. So here we have a nice little figure 2613. So this is just a chart that's showing us our evolutionary time and species over the course of that time. So we have time that's increasing, it's going to the right hand side, and it's going up here. The number of, or the evolutionary time is going to the right on the bottom here. The base differences, or the number of changes in your genetic code, are on the vertical side, so going upwards. So the higher that a species is positioned, the more genetic base mutations they have, and the farther over a species exists is the younger that they are. Okay, so you can see here we have about 6 different species plotted on this graph. So as time is going on, we're getting more and more mutations occurring. So these are the base differences in your DNA strand here. So you can see that if you have a data point that's older and has less base differences, typically they show, if there's 2 species at this plot point, typically you can see that these two species have a common ancestor, and they don't show a lot of base differences in their genetic code. But if you have a species that's very new and has a lot of base differences, or if you have two species in that area, then you can say that they have a distant common ancestor and show many base differences because they've had a lot of time to change over the course of that time genetically. And we can see this in primates. And so with primates, we have different genes here that we're going to analyze real quick to study this phylogeny and kind of evaluate the timing of this evolutionary change. So you can see the ancestral species at the nodes here, or the areas where the phylogenetic tree is diverging. And we have ancestral species A, B, C, D, and E. But you can see at the branch tips here, we have the species that were descendants of these ancestral species. So we have different types of monkey species, orangutans, humans are up there, and chimpanzees, gorillas, and another sort of tree monkey, the samangs. And so you can see that over the course of time, the individual species that have occurred more recently typically have more base pair differences than the evolutionary ancestors here. So, for example, the common ancestor E, or the primitive ancestor E, that created the regular common chimpanzees and the pygmy chimpanzees, those two species have way more base differences, and that common ancestor has way more base differences than you would see in the primitive ancestor species A, because they had a lot more time to change their genetic code. Now, say we have primitive ancestor number C, or letter C here. It's probably equally related genetically to primitive ancestor E and primitive ancestor A, because it's been right around the middle amount of time that's happened 10 million years ago, and out of the 25 million years ago where the species is plotted, it's right around halfway. So they're probably equally related, and the base pairs are probably the same amount of difference as the newest primitive ancestor and the oldest primitive ancestor that we have. And we'll see this again most likely in your Module 9 quiz. So that's all I really have to talk about in terms of molecular clocks. It's just basically the whole point is trying to explain that your genetic code can tell us, where you might fall on the evolutionary charts, just because those genes do change over time, and we see that happening in a lot of morphological features as well, the morphological features, but they're linked to the genetic features. So. Okay, so let's talk about horizontal gene transfer here. So we're familiar with vertical gene transfer. You all have seen this before. This is where your DNA or your genetic code gets transferred from your parents, your mother and father, down to your offspring. And so you have, chromosome from your mother and chromosomes from your father. And this is where we're getting that original vertical gene transfer. So y'all are familiar with that. So what about horizontal gene transfer? So another name for horizontal gene transfer is also called lateral gene transfer, because it moves laterally instead of vertically. It's the same thing. So if you see lateral or horizontal gene transfer, those two are the same exact things. It's just two names for the same thing. Okay, so we're familiar with vertical gene transfer, so what about horizontal gene transfer? What is it? So horizontal gene transfer is the passing of one or more genes through routes other than from your parent to your offspring. So This is going from one organism to another, typically not parent, not offspring. And we see this happen in a few different ways. So in these images here that I've included, these are just some bacterial transfer, gene transfer that you might see through a few different avenues. So the one image shows bacterial conjugation, where a bacteria actually connects to another bacteria that's completely different, and then they share some genes or some DNA that way. And another thing you might see too is bacterial transformation, where a bacterial cell is ruptured and some of its DNA ends up on the outside of the cell, and another bacterium comes over that's completely different and ends up collecting or obtaining some of that DNA and taking it into its organism. And so this is just some weird ways that these organisms will get unique genes that is not passed on from parent to offspring. And we see this really common in bacterium and certain unicellular eukaryotes. So typically it's more common in these particular organisms, and it's more rare in things that you might see like fish or fungi or raccoons, you know. The multicellular eukaryotes don't typically have this happening, but in bacteria and other unicellular organisms, it is a common way for genes to move around, or DNA to move around. It is typically rare in multicellular eukaryotes, so you typically won't see a bat sharing DNA or genes with a frog. Although it does happen, it's not completely impossible, and this can be through other means that occur, typically through bites or some sort of other way that these multicellular organisms come into contact with one another. But typically it's rare. We typically don't see this. And so this kind of indicates to us that instead of a tree of life that we have, it's really more of a web. So you'll see, instead of it just growing up and out, you'll see that genes are transferred across and horizontally as well. But it is more common in that bacteria, archaea, and the unicellular eukaryotes as well. So it's really more of a web. Everybody's interconnected, or a lot of species are interconnected. But you can see in this particular web of life, the animals, the fungi, the plants, the algae, and some of the protists typically are not privy to that horizontal gene transfer. So that's why their branches are kind of spaced out like that, and they don't really have a lot of horizontal or lateral interaction. All right, so that's really all I had. Those were two really short sections. So that's all I had for those two sections of our Module 9 here for taxonomy and systematics. So I do have a quick review, so you guys feel free to hang out for it. It might help you with your quiz or make your quiz more streamlined, hopefully, or you could just, if you like to just listen to me talk, that's fine too. So this is what I would normally do, you know, in the classroom, but I miss you guys, and so I've included just a little review here. All right, so these are examples of a clade. True or false? So I have two examples here, the highlighted area in blue, the highlighted area in green. Are they a clade or are they not a clade? So these are a clade, yay! So they are encompassing all of the descendants and the common ancestor for those descendants. Not all the way back to the original common ancestor. The clades can include a recent common ancestor. They don't have to include all of them, but they want to include the recent ones and the descendants. Okay, so you might see something like this on your quiz. So if you remember the principle of parsimony, which is the simplest or most logical explanation is typically the one that's correct. That's the principle of parsimony. Which species has a recent common ancestor with the nucleotide sequence AATTC? So we have 4 options here: AAATC, ATTCC, AACCG, and TATTA. So I'll let you guys think about it for just a second here. Alright, so what's the correct answer? Correct answer is AAATC. Why is that the correct answer? So if you look at all of the base pair nucleotides here, you can see that there's only one that has changed from the original primitive ancestor. So the only base pair that has changed is the 3rd base pair. So that T had just been changed to an A. So likely, since that's the most simple and logical explanation, it's the most likely to be right. So that one is likely to have a very recent common ancestor because the amount of changes are not exorbitant. And so the other options that I've listed here have 2, 3, or 4 different base pair changes. And typically you'll see that with a very, very distant common ancestor. So if the question was asking you for the most distant common ancestor, you'd pick the one that was the most different, which in this case I believe is the 3rd one here, which would be 3 base pair differences, which is AACCG. Okay, this question here, considering the principle of, oh, I didn't change this one. Okay. The question that I'm trying to ask here is, if you look at this highlighted clade here, the reptiles, so the reptiles in this, this clade are including turtles, lizards, and snakes, crocodiles, and birds. What sort of description could you use to describe this clade here? Monophyletic, paraphyletic, or polyphyletic? So just ignore the question there. I forgot to change it when I was making my PowerPoint. The answer here is monophyletic. So that's because it includes all of the organisms at all of the tree branches and the common ancestor. It's all inclusive on this in groups. So if we were excluding a species, then we would be paraphyletic. If we were polyphyletic, we would include species that did not have the same common ancestor. So hopefully that makes sense. Sorry again about this question not being changed correctly. Okay, so I changed this question correctly here. So what species did the ancestor D that I circled here in red and give rise to? So the options here are humans, common chimpanzees and pygmy chimpanzees, all of the monkeys, or humans, common chimpanzees, and pygmy chimpanzees. So this I felt like was a pretty easy question. So what do you all think? Correct answer is the last one. So the ancestor D species gave rise to humans, common chimpanzees, and pygmy chimpanzees because it's a common ancestor for all of those branches that are coming off of that ancestor node there for species D. Okay, is the blue shaded area considered an outgroup or an in-group? So you can see there's like a yellow shaded area, but there's also like a blue shaded area. I'm looking at the blue one, B, C, and D. That's all highlighted there. The correct answer is in-group. So if we would be talking about a group of organisms or a group of different species here, and we circled all of those, that would be considered our in-group. And species A here would be considered our out-group if we were talking about those specific ones that have happened more recently in the evolutionary timeline. Okay, so which colored circles are considered a synapomorphy? And you can see that the circles here, the white circles are a derived trait, the black circles are an ancestral trait. And so which one is a synapomorphy? The answer would be the white circles here. It's a specific trait that is a derived trait that comes from a common ancestor that is derived in a species, 2 species, 3 species, however many are coming off the clade there. And typically you'll see that that's coming from a common ancestor that's slightly different than what you'd see in the other species on that cladogram or that phylogenetic tree. Okay, so what is a phylogeny? So make sure you know your terms. That's another good thing to keep in mind. Make sure you're making note of every term that we're talking about, because there's a few here that can get a little confusing when we're talking about cladistics, phylogeny, taxonomy, systematics, all of those terms. Just make sure you know those. So a phylogeny is the evolutionary history of a species or a group of species. Simple enough. Remember that typically we see a phylogeny or a phylogenetic tree that's indicating time in our chart here. And the length of the branches typically indicate more or less time that has passed, whereas a cladogram we typically see the interaction of the evolutionary relationships between species, and they're all pretty evenly spaced out, and they don't have branch lengths that indicate differences in time necessarily that has passed. So what are cladistics here? Cladistics are the classification of species based on evolutionary relationships. So we're looking at the relationships that have occurred between species that are on a chart here when we're talking about cladograms. We're looking at, derived characteristics or traits, ancestral characteristics or traits. Who is the most recent common ancestor? Who is a more distant common ancestor? Who is most closely related based on the common ancestors or on the descendants that we have circled in an in-group or an out-group? So you might see those kind of questions on your quiz. Okay, here's a good one if you guys can remember. So what is the correct order of taxa used to classify organisms? So if you remember all of the taxonomy is the classification or naming of species. So what is the correct order? So you can go in this case from largest to smallest. So the most broad order or taxa all the way down to the most specific group of taxa. So the correct answer for this one is, oh, you can't see it because I moved it behind my screen here. Oh, that's unfortunate. Okay, so the correct answer, you'll just have to listen, is the domain, the largest group. It encompasses everybody, all the bacteria, archaea, and eukarya. Then we have our kingdom, which is next smaller. Then we have our phylum. Then it's our class. And then it's our order. You can't quite see that behind my my little outcropping of my video here, but it's class, then order. Then we have family, which is also behind my screen. And then we have genus, and then we have species, or the specific epithet. And this was this image here was just classifying a fox species. So that's why it has the actual classification in this image of Eukarya, Animalia, Chordata, Mammalia, and all the way down to the Vulpes. So this is just an example here, but this is the correct order of taxa used to classify organisms. Okay, so what is it called when one species that we have, an ancestral primitive species, let's say, in this case it's some sort of birdfinch, what is it called when that one species diverges into 2 species or 3 species or 4 species or more than that? So what is it called when that one species diverges into more than one species? So we have anagenesis, cladogenesis, node, in-group, and out-group. So this is where you place your bets. And the correct answer is cladogenesis. So cladogenesis, if you recall, is the diverging into more than one species. And the opposite of that, or I guess the opposite of that, would be the anagenesis, which is one species changing into another species or evolving into another species. So we have the divergence of species, cladogenesis, and the morphing or the evolution from one species to another, and that is our anagenesis. So the anagenesis is a very vertical line. You just see from one species to the next species. You don't see a divergence. You only see that in your cladogenesis. Okay, and I believe this is my last slide for you guys today. So nice, quick, easy lecture for a Friday. So what is the specific epithet or the species name, just the species, for the hooded merganser, otherwise known as Lophodites cucullatus? So take a second to think about what I'm asking here for. I'm looking for the specific epithet or the species of the hooded merganser, Lophodites cucullatus. So my options here are Lophodites cucullatus, regular cucullatus, Lophodites, and cucullatus spelled a little bit differently. So make sure you're looking at how I've written the spelling as well, because remember that binomial nomenclature and how we write our binomial nomenclature is important, and you'll see that in your quiz. So keep that in mind on how we are writing those. Okay, so the answer here in this case is cucullatus. So that is just the specific epithet. That is the species name. If we were asking for genus, then I would put Lophodites. And then if I were asking for the genus and species, then I would put Lophodites cucullatus. And make sure you're spelling it the right way. Remember, when you're writing using binomial nomenclature, you're capitalizing the genus name, Lophodites, and then the second part of the word, cucullatus, is lowercase, but make sure to remember that it's italicized, the whole word. And if you're handwriting it, then it's underlined, because you won't be able to tell if you're handwriting it, if it's italicized or not. So I believe that is it for you guys today. I hope you enjoy your weekend and enjoy my lecture series, and I will be posting more lectures and labs here very soon. So thank you.